Holly must be where it's slow. It's taking her a minute to get in.
Oh,
Okay. Welcome everyone to our rounds for December, the last one for 2025. Today we have sorry, I'm going to mess it up already. Pekka Määttänen. He is currently the associate professor and chair of biology at the department at Berman University. He holds a PhD in biochemistry from McGill University and an undergraduate degree in cell and molecular biology from the University of Toronto. Sorry, Pekka, but I'm not going to get into some of these words because I don't know how to pronounce them. But he has recently turned his attention to haskap berries, and I hope you have a picture because that's not as common in Ontario, and whole ground flaxseed impacts on the cardiometabolic disease and the impacts of lifestyle medicine intensives on the microbiome. He recently spearheaded the first lifestyle medicine and flourishing symposium in central Alberta in May of this year. He is happily married to Sarah and has 3 children who love nature, and he's hoping they will love biology as much as he does. And he's open to questions and answers at the end of the presentation, and you can put them in the chat as well, and we'll answer them at the end. Thanks. Welcome.
Thank you so much for the welcome. Everybody hears me okay, I hope. Yes, my name is Pekka Määttänen, and I'm just excited to be here to be able to talk to you a little bit about optimizing gut microbiota with diet. So I'll jump in pretty quickly here. This just first item of business, I really don't have much disclosures to say. I've done one food-based clinical trial with the Weston Foundation recently. So other than maybe pushing haskap berries at you, I'm not going to do much else. And yeah, I don't have anything else to disclose. Okay, so our objectives today are to recognize the difficulty in defining a healthy and unhealthy microbiota. And I want to emphasize that, the microbiome field has been an area of much hype. And I'm not saying all of it is unfounded, but some of it is probably worthy of some skepticism. Let's put it that way. So I just want to give you sort of a healthy, balanced view of, what we can say from the microbiome side and what we can't. And then also look at, so that'd be my second point there, appreciating the gaps in understanding the gut microbiota and its variability in particular, how different it is from one person to another. Understand influences of dietary components and metabolites produced by microbes. So this is an area where I think we have a lot more evidence, and we can really kind of focus in on some of the major benefits of some of these metabolites that microbiome or that the microbes in us produce as we consume different foods, and then list some of these microbial metabolites that have evidence-based effects on physical and mental health. And then finally, identify dietary strategies to optimize metabolic outputs of microbes. So, optimizing your kind of the title, your microbiota with diet. So with that, some definitions. A lot of people use interchangeably this idea of microbiota, microbiome. What's the difference? So microbiota these are the trillions of microorganisms, the bacteria, archaea, fungi, protists, even viruses found in and on the human body. Vast majority of them are in our gastrointestinal tract, as I'm sure you're all aware. Whereas the microbiome would be the sort of holistic genetic content of these microbes. So keeping that in mind, often we use microbiome interchangeably with microbiota, but just, for definition's sake, to know who are the microbiome or what are they. So primarily the bacteria and the archaea, when we look at them, we have about 38 trillion cells. And this kind of fluctuates up and down, but depending on the person. The gut microbiome remains incompletely understood. So we have about 3,600 high quality genomes in this bacterial archaeal area that are can be mapped to really specific species. But that leaves about 17% of the sequences that we identify of the microbiome sort of unannotated. So there's a lot of dark matter there. We don't really understand all of them. And certainly that's an area of active research still. And I would argue that there's probably a lot more there that still need to be figured out. As the tools improve, we'll get better at characterizing them. So when we look at the other, I would say perhaps more minor, but also very influential members of the microbiome, we have the fungi, which make up only about 0.1% of the total species in the gut, at least. And you have Malassezia, you have Candida, you have Saccharomyces. These are all part of this, mycobiome, we could call them. These fungi involved in living in and on our bodies. There are also protists. Now, we don't typically in developed nations have as many of the protists, but certainly they can compete with bacteria for energy sources and have some influence, but they're more prevalent, as I said, in less industrialized populations. Viruses, interesting if you ever want to hear more about viruses and the virome. There's a guy by the name of Colin Hill from University of Cork, Ireland. Listen to him, some fascinating work he's done. But essentially, there are many, many, many of them. Think of them as, the flu of the bacteria, if you will. They're the ones that are really affecting these bacteria, and their populations could be dramatically impacted by, phages that are present in the gut as well. So lots and lots of them still being understood, many, many of them. For the purposes of this talk, we'll primarily be looking at bacteria, but just to give you a bit of a sense of, this has been changed over time, but this idea of, genetically, are we more human than microbe? Well, actually, we are more microbe than human genetically. There's about, 2 million, even up to 20 million microbial genes present in and on our bodies, whereas our own human genome is only about 19,500 genes-ish, depending on how you define them. Cell number, though, it was kind of touted back in the day that, we were outnumbered in cells by our microbiome. That's not necessarily the case. It's more similar. We're about, 4 times 10 to the 13 microbial cells versus 3 times 10 to the 13 human cells. So they're similar amounts. You could argue that if you look at my funny picture here, humans still wear the pants in this situation. That they're still very important. And of course, microbial cells are much, much smaller, about 50 times smaller than a human cell, depending on which cell you're looking at. Okay, so that's why we don't look like a giant walking chunk of stool, if you will. All right. So looking at what makes us unique, this is the part that I think is very important for us to consider. Our human genomes are very similar from one to another. So we're about 99.9% the same. We really, and this allows easier comparison from person to person. But when you look at microbiomes, this is where we have a lot of variation. So about 80 to 90% different from one person to the next. And this doesn't mean that we're going to be, like completely different in terms of, what species you may or may not have. Some of the major phyla may be the same, but there are a lot of differences from person to person. And I want to drill this point home because I think this will help us understand what we can apply when it comes to the microbiome and its understanding to things like healthcare, because you have to think about it. If I don't know what a healthy microbiome is, because there are a lot of healthy people with very different microbiomes, then how do I define that, right? How do I actually say these are the healthy ones or those are the healthy ones? And that is a challenge. So as I was just alluding to, because people are more similar genetically, it's possible to compare genes. We can say, okay, there's this genetic disorder, there's this particular, change in gene expression in a certain disease and so forth. Whereas when we look at the microbiome, because it varies so significantly, we have to start to think, okay, well, wait a minute, maybe this has to be even more personalized, even though we're, genetically, we're talking about personalized medicine. Well, what about our microbiome? Well, there are things like fecal microbial transplants that seem to work, but that's a whole ecosystem transplant that's happening. That's a pretty big shift that occurs, and you're not just changing one thing, you're changing everything kind of at once. And so that may work, but trying to tailor, trying to tweak one person's microbiome just with one or two microbes may not be as easy, although there is some evidence that probiotics can help. So this kind of takes us to the place where we could talk about what is a healthy microbiome. And I don't actually think we have a good definition of that. We do have some clear ideas of, what behaviors we want our microbiome to be engaging in. And we do have some good ideas that a diverse microbiome seems to be beneficial, although not always, because you don't want to have more bad guys, right? And then who are the bad guys? There are certainly some microbes, as I'm sure you're well aware, C. diff and some of these microbes that we really know are not great to have in overabundance, EHEC and so forth. But we do not really have a clear definition of that healthy microbiome. And so one thing that I thought to do just in some research myself, which some of it's a bit redacted here because I haven't yet published, is what if we look at a person who's unhealthy and that same person, we help them achieve a healthier status, whether it's cardiometabolic health or mental health or whatever. We move them into that healthy place and we monitor their microbiome from sickness to health. So we check, okay, when they're sick, this is the microbe, this is the population they have. And now in that same person, using them as their own sort of internal control, what's happening to their microbiome? Is it shifting? And if so, how? And that was kind of my thought of, okay, a personalized approach. So we applied this in sort of this is actually a program called the Karpala program in Finland, where we apply a broad set of principles of lifestyle medicine, nutrition, exercise, water, sunshine, temperance, fresh air, rest, and trust. Okay, so that's a big mouthful, right? But 8 main principles. And then that program allows for these people who are primarily pre-diabetic or type 2 diabetic, overweight, cardiometabolic disease, a lot of improvements. So we see significant improvements in their BMI, their blood pressures, their LDL cholesterol, that sort of thing. So what's happening to their microbiome? So we took some stools from these people, and not surprisingly, though I'm not showing it here, I would have had a nice bar chart for you if you have the presentation yourself. You can see it there, but essentially, everybody looks very different in their startup microbiome. But do they all become the same at the end? No. They just all shift in their own way, not surprisingly. And so what I think this drills home is that we are seeing shifts in an individual person's microbiome in response to a dietary change, but not necessarily identity change. So not all the microbes change to different microbes. And I think this is somewhat analogous to when you as a person start, eating healthier. You don't become, I don't become Milan or somebody else, right? I'm still me, but I've changed. My physiology has changed. The metabolites I produce have changed. Those things have changed. So I believe microbes, actually, some of them can go through a similar process. Other ones will, obviously, they'll be starved because they're not getting the foods they want. Maybe they want constantly mac and cheese and burgers and white bread, right? So they're a little different. And so there will be a bit of a starvation effect for some of them, but I don't believe that we see necessarily this holistic, like whole ecosystem change. We see a gradual shift towards different metabolites produced. Similar approach we did looking at, so this was this species variation that happened. I did one looking at mental health. So there's a 10-day residential similar program, very much looking at depression and anxiety. We see improvements in depression scores and anxiety scores, EQ improvements. What happens to the microbiome? Well, we don't see any obvious clear trend across different people, but we do see changes within people, and those changes, even them at the level we're looking, because we're just looking at who's there, doesn't really show us much. So maybe we need to look at something else. And this is where I I would have shown you some nice bar charts, but basically you just have to take my word for it that we saw shifts, but everybody was different from one another. So maybe instead of trying to look at the genetic identity of these microbes or their microbiome, right? Maybe we need to look at the metabolites they produce. What are they actually doing in our gut? So just like you or I could be restored to health, as I was mentioning, perhaps our microbes can adapt to a new, healthier environment, maybe different nutrients available. They could undergo their own behavior, lifestyle change, and they're having a little kind of lifestyle program in your gut sort of thing for themselves. So this makes us or begs the question, well, how do we establish our microbiome? How is a healthy gut established? Because I think that's a key point. If we can say, okay, these are the key steps towards developing a good, healthy microbiome, then maybe we can have a better handle on how to maintain that. So when we go back to our early days, if you will, passage through the birth canal, that's when you first get seeded with your microbiome from mom. And the baby does enter a world teeming with microbes. And as soon as they're out, of course, they want to go and drink some breast milk. And that is important, not only to provide that, that passive immunity, but also the contact with mom, the contact with microbes present at the nipple and so forth. And also in the milk itself, we know that there are lots of good things. Well, what are some of those good things in the milk? Well, one thing that was discovered very early on, you'll notice on this slide, this is very, cutting edge research from 1900, right? Where it was found that there was this Y-shaped bacteria, they call them bifidobacteria now, that were found in these more acidic stools that infants that were breastfed had. If they were fed a cow's milk supplement, their pHs were a little bit higher, so they're more alkaline, and they also didn't have quite as many of these bifurcated bifidos. And infants lacking this bifidus dominant microbiome, if you will, were more likely to have some gut-borne pathogens. So this was back in the day, they already knew this. So when we look at, this brings us back to the definition of what's a prebiotic, what's a probiotic. I'm sure you're all well aware, but just to kind of give you the canonical definitions. So according to the International Scientific Association for Probiotics and Prebiotics, a probiotic those are the live organisms that, when administered in adequate amounts, confer a health benefit to the host. Prebiotics are a substrate that's selectively utilized by host microorganisms, conferring health benefits. So the prebiotics feed the probiotics, if you will, but probiotics, can be administered and could provide some health benefit. They're living, ideally, right? They need to be living, whereas prebiotics are the foods we eat. Now, one important thing I want you to consider, and I know you learned this probably in your micro classes many years ago if you took them, is, one of the first steps to sterilizing, I don't want to say sterilizing, but removing some potential pathogens in the foods we eat is in the low pH stomach, right? Our pH in our stomach is very low. It actually is not very conducive to growth of a lot of bacteria. In fact, it kills many of them. So if you're taking in bacteria that are not encapsulated or are not protected from that, they're not going to get past that to the gut as readily. Whereas if you have a population of bacteria in your colon that are receiving fiber, which we're going to look at very soon, foods those prebiotic foods can support the growth of those bacteria that are already present down there, and you're not having to, repopulate that constantly. Of course, after having a course of antibiotics, it could be pretty important to do that. So just keeping that in mind, that sometimes, if we take a probiotic that's not protected from our stomach acid, it may be challenging for it to actually make it there to where it needs to be active. So when we look at prebiotics, in mother's milk, this is something that not too many people are aware of, but of course, you as physicians, I'm sure, are well aware that there are fibers in human mother's milk. These are non-digestible oligosaccharides called human milk oligosaccharides. And there's quite a large proportion of them. There's also quite a bit of fat, so our lipids, including these pre-processed omega-3s that dampen inflammation. So they're pro-resolving lipid mediators found there. And then protein, lactoferrin, immunoglobulins, and so forth, lactose, quite a large amount of that. Probiotics as well, and then even some microRNAs and stem cells. So there's lots of good stuff in human mother's milk. Very difficult to replicate in a formula, but the prebiotics are being replicated now. You have the SC phos and the GOS and so forth that's included in many formulas. So there's a lot of diversity in human milk oligosaccharides, just to give you a sense of how broad the structures of these are. Very many different types of them. And they, just like, if you look at their shapes here, there's just so many different structures present. And this is already found in human mother's milk. So there's this great diversity of non-digestible to us fibers found in human breast milk. Well, that's an interesting thing. What about when we go on from there to other foods? So what do these human milk oligosaccharides do? They feed the good bifidos. So those bifidobacteria can then exclude, through competitive exclusion, they can help to prevent overgrowth of some of the more pathogenic types. They induce expression of mucins. So we found that actually the HMOs on their own can actually increase expression of mucins. That was back at SickKids when I was doing a postdoc there. They induce expression of tight junction proteins as well to help zipper together your intestinal epithelial cells. So you're, less likely to have that leaky gut. Induce proliferation of intestinal epithelial stem cells. So you have a more, beefy gut layer there as well. And then modulation of immune function as well. And we'll look at that when we talk about fiber. So dampening inflammatory reactions in the gut. And then some of the sialylated HMOs have been shown to have roles in brain development, cognitive function, synaptic transmission. So they're pretty important for a lot of things. So just to kind of follow through this process of how our microbiome develops. So most of our microbiota is present in our gastrointestinal tract, and there's about 3,600 different species that have been identified so far. They interact with us through this large surface area of our gut, about 344 square feet, probably the size of a smallish apartment. And the human milk oligosaccharides, including more than 200 different non-digestible sugars, they feed these bifidobacteria, and the bifidobacteria, as a result, actually produce short-chain fatty acids. And that's one of those metabolites, those key metabolites that we're going to touch on, has been demonstrated to have many, many benefits for us. And by about age 3, we see stabilization of this microbiome in each person. And if you have these bifido sort of rich fiber fermenting type of microbes present, then that can help prevent some autoimmune diseases. Now, that's at least been correlated. Now, if we look at dietary fiber when we are weaned, well, this is where having a variety of different fibers, remember we looked at the variety of different HMOs present in mother's milk. Well, you need a variety of different fiber foods to fulfill that requirement for those healthy fibers. And meat, on the other hand, is fiber-free. So that's one thing, most people look at meat and they say, oh, the red meat itself, it has its problems because it has the cholesterol and the, but I'm looking at it now from the perspective of what it doesn't have, right? It's lacking the fiber. And it also does contain some of the things that we know, for example, are going to induce some toxic metabolites from the microbiota. So deoxycholic acid is one example. We'll look at that a little bit later. So when we talk about weaning, this is actually a picture from Finland of a bowl of cereal I was having in the morning there. You can see that when you're eating food, of course, you're transitioning the baby from this, maybe mixed breast milk with some other foods. You start to add some foods that are going to hopefully contain some fibers in them. You're not going to throw in, nuts right away, but you're going to maybe puree some food and so forth. But you want to include lots of fibers in those foods. And the key there is you're trying to continue to support those healthy microbes present in the gut of this baby. And what are some key things here? And I mentioned pureeing. Well, if you think about fiber from plants, what fibers are we talking about? Because remember, human mother's milk isn't like this, isn't like celery, right? It doesn't contain like big chunky fibers. It's going to contain these soluble fibers. So how do we access soluble fibers from plants? Well, we need to chew our plants well, or they need to be masticated. They need to be crushed. They need to be maybe blended. And so obviously chewing is really helpful. Why? Because it breaks open the plant cell walls and allows release of some of these resistant starches, some of these galacto-oligosaccharides, fructo-oligosaccharides. So chewing our food is key. I know my grandpa used to say, chew 10 times before you swallow. I don't know what the number is for the secret number is, but making sure you chew your food enough so that you release those soluble fibers from inside the plant cells so that they can be accessed readily. And there's so many different types. Again, it's an area of much research still. In fact, we know too little about all the different types of fibers in foods. I think that's an area that needs more research. So when we compare, just looking at Mediterranean high fiber diet compared to, let's say, our Western high fat diet with emulsifiers, you can see that there's a couple of benefits here. So as the fiber is taken up by the bacteria will release short chain fatty acids, those SCFAs, also some secondary bile acids, lower levels of endotoxins. And what will happen is, notice the mucus layer here is thicker. This is a key point. This mucus layer stays thick. And though this image doesn't show it, there's a mucus layer also that's closer to the intestinal epithelium that is mostly sterile. So it stays pretty bacteria free. Now it's not 100% free, but it's pretty free of bacteria. And that upper layer, lots of bacteria within it. And they like to live there and they get their fiber in your diet and they're happy. But if you start to remove fiber, I like to put it this way, it's kind of simplistic, but the bacteria, they need food. And so they might start eating mucins because mucins, guess what? The mucins are made up of some of these carbohydrates that they like to consume. And so they will consume them. Now, not all not all microbes are mucolytic, but some of them are, and they'll start eating away at this mucus layer. And now you get down to that closer and closer to the epithelium, and you start to have leakage into some areas here. The other issue is because you don't have short chain fatty acids, you don't have dampening of the immune response. So the T regulatory cells are actually upregulated by short chain fatty acids. We know butyrate in particular is a strong upregulator of Tregs, and that induces this, dampening of your Th17 response, and you have this less aggressive inflammation in the gut. Whereas if you dampen the Tregs, now you have, higher Th2, Th17 going on, more gut inflammation likely. So this is kind of one of the theories of why this Western diet, and I will add, notice it says emulsifiers here. So I don't know if any of you are much aware that there's been some studies on carboxymethylcellulose or polysorbate 80. These are some of the common food additives found in ultra-processed foods. You'll find them in a lot of times in like ice creams and things like that. Now, those actually have a very specific effect been shown in even some clinical trials in people, but initially in mice, where these carboxymethylcelluloses, this is work coming out of Benoit Chessieng's lab in INSERM in Paris. Basically, what they did was they fed this emulsifier, and it causes the microbes to become more invasive. They're not really sure why, but it seems to, I actually had a talk with him. I asked him, hey, what do you think this is happening? And he said, well, I don't know. There's maybe a receptor on the surface of the bacteria that, binds to this, and then it induces them to start expressing massive amounts of fimbriae and flagellin. So they become like these angry bacteria. And they attach much more likely and invade much more likely. They found this with the AIECs in particular, adherent invasive E. coli. So what he showed in people is that this also dampens short chain fatty acid production for some reason, just eating more emulsifiers. So just a very, very interesting thing to consider. Okay. And this is, sorry, I should have just shown this slide. This kind of touches on that, that study that I was just talking about, where you get more adhesion, more invasion with these dietary emulsifiers. And actually, there's an upregulation of these flagellins and these adherent molecules on the surface of the bacteria. So going back to butyrate, I like to call it the beauties of butyrate, because butyrate is really fantastic. It has so many benefits that have been demonstrated. So this is one of those metabolites that we wanted to look at, the evidence-based metabolites from microbes. Acts as a source of energy for colon cells. So colon cells are one of the few cells in our body that outsource their metabolism to microbes. Microbes convert the fibers into butyrate. The colon cells take up that butyrate and use it as an energy source. And this energy source, continuous use, you might wonder, why do people who eat so many fibers, have a much less likelihood of having colon cancer, though it's not 100% right. There are genetic predispositions, but there's definitely some benefit there. It's because the butyrates actually push those colon cells into an area of metabolism that isn't really compatible with cancer. And there's been lots of mechanistic studies to show that. So stimulation of IL-10, I mentioned that already. So dampening inflammation, promoting Tregs, decreasing expression of genes involved in inflammation, stimulating expression of mucins, immunoglobulin A. So, not only do these fibers say, hey, we're going to feed the bacteria so that you don't eat our mucins, they also upregulate production of mucins. So you have this, sort of double whammy that's happening. And then brain inflammation has also been demonstrated to be dampened through butyrate. So how much fiber does a person need to eat a day is kind of a contentious issue. The Canadian nutrition guidelines will say that about 28 grams a day in adult men, or for women, it's 28 grams a day. For adult men, 35 grams a day. I think we can safely go higher than that. I know 40 grams is it's not out of the question, but gradually moving in that direction, right? Because obviously, you've probably all seen this. If you try taking fiber too quickly, maybe in your patient or yourself, it gets out of hand pretty fast. So you need to gradually work your way up. But staying hydrated is really important as well. But I would argue that back when we were used to eating fiber, and we were having this discussion actually just before we got into this talk, it could have been up to 100 grams a day. And there's some studies saying that that's about how much they used to eat, more when we were actually, gathering our own foods and not processing them too much. So notice in Canada, we're actually worse than Americans in terms of our fiber intake, about 14 grams a day versus 15. I know it's not that huge of a difference, but so we really need to get on ourselves about this and make sure that we get enough fiber. So what fibers are really good? So we call them the microbiota accessible carbohydrates, or MACs. And they've been demonstrated, these soluble fibers, to have lots and lots of benefits. Which ones are they? So short chain fructooligosaccharides, or SC-FOS, GOS, galactooligosaccharides, inulin. Where do we find them? Garlic, onions. I don't know if you've heard of Jerusalem artichokes. These are a little over the top. They're like I think pound for pound, they're the most rich source of inulin possible. But there are so many other whole plant foods that contain fibers. Pretty much any plant has fiber in it. It's just, take your pick, right? And I used to actually run a program for weight loss. It was called the Full Plate program, or full plate diet, but now they've changed to Full Plate Living, and it's actually free online. So if you ever want to look into that, it's called Full Plate Living. And again, I'm not, I don't have any shares with them or something, but it's free. So essentially, I don't know how they could make money. This program is basically a way to enhance fiber in the diet of people, and it's kind of a self-directed free thing where people can just go on and say, hey, I want to eat, I want to lose weight, I want to be healthier, what do I do? And it gives you a fiber guide where it tells you, okay, these are all fiber-rich foods, and then you just go and pick your favorite foods and you just eat more of them. So it's not like, you have to prescribe, okay, eat this fiber-rich food. The patient can pick their favorite fiber foods and go for it. There's enough options there. So when it comes to plant fibers, I wish we knew more. Actually, surprisingly little is known about the chemistry of some of these plant carbohydrates, partly because they're really challenging to study and there's just a lot of complexity there. So there's many, many thousands of them in the catalog. UC Davis has started this. They call it the Davis Food Glycopedia. They're trying to, create this large scale catalog of them. But the prebiotics really focus on short chain fructooligosaccharides, inulin, galactooligosaccharides. These are the ones that, you see them fortifying like milk, like the formulas with. Resistant starches are also really good sources for microbiota accessible carbohydrates. And their degradation is pretty complex. It's pretty fascinating, actually. Like if you take pulses and depending on the way you prepare them, can actually affect the types of starches and types of resistant oligosaccharides present in there. So if you take the intact pulse cells, you can get these RS1 types. Then you have these native pulse starch, RS2 and RS3, and then there's crystallized starch granules or retrograded starch that can happen. So there's lots of different ways that these different fibers can appear. And again, all these benefits that they have. This is kind of a neat thing to think about. The fiber, if you think about the ecosystem of bacteria in your gut, they will actually eat at the bits of fiber in you, and they actually cooperate with one another. So some of them are able to, access some of the larger chunks of fiber, whereas other ones need the pre-processing by some microbes in order for them to be able to access those carbohydrates. But just to see kind of the packaging, it's pretty neat how amylopectin and amylose are involved here, and you have this packaging into these starch granules that are quite efficiently found inside. And just to reiterate what I was saying, you have this food chain, if you will, or food web for fiber, where you have primary degraders growing directly on the resistant starch themselves and start to penetrate the surface. And then they start to break things out there, and then you have these secondary degraders and then cross feeders. So the secondary degraders, while they can grow on starch by themselves, they actually don't degrade this resistant starch very well. So they need to kind of get the help of the primary degraders, if you will, and then your cross feeders. They can't grow on starch, but they can utilize the byproducts from the others. So there's this ecosystem happening, and that's where you got to think about, like, okay, if I haven't been eating a lot of fibers, maybe I need to gradually beef up, my bacterial ecosystem to the point where now it's in a place where it can do the things that it was meant to do. And it's actually pretty slow for this all to happen. And yet the effects are really, really beneficial for our colon cells. Notice they nourish the colon cells and provide those anti-inflammatory effects. One thing I wanted to say is, think about the colon. You have along the length of the colon, you can have a sequential series of metabolic steps happening for these fibers, where later on, and this is where studies I think are going, where we're looking at different regions within the colon, the distal colon versus proximal, like where are these different fiber fermenters found. You probably later on, even the mucus layer starts to change, where you have a thinner but more resilient and slippery mucus layer in the distal colon, whereas it's a little thicker and less, less dense at the closest part at other parts of the colon. So there'll be differences regionally in our microbiome along that length. Now, how about pathogens? Can we actually demonstrate that if we add fiber to a diet, that it can influence, pathogen clearance? Well, looking at C. diff in particular, this was shown in a mouse study, but essentially you have these microbiota accessible carbohydrate deficient diet versus a fructooligosaccharides enriched diet or an inulin enriched diet. And you can see that in the inulin enriched diet in particular, after infection, you have much more rapid clearance of your C. diff than in the case of the deficient diet. FOS didn't do quite as well, but it did have some benefit there. So it's just an interesting thing. So key point here is don't starve your microbial self. Keep feeding those good bacteria. One thing we found, though, in addition to the effect of the fiber on the bacteria and then the production of short chain fatty acids and all the benefits that I talked about, it seems like fibers can actually directly benefit the intestinal epithelial layer. So these were some studies we did on Keiko2 cells grown in monolayers in dishes where we gave them EHEC either in the presence or absence of these fibers. Now, in the control case, so enterohemorrhagic E. coli, what's happening here is it's breaking up the tight junction. So you can see these green, these are the tight junctions that we labeled Z01 and so forth. If you give EHEC, they damage the tight junctions and the intestinal epithelium is really compromised. And you can check this with fitzy-dextran and so forth to see will it penetrate. If we use inulin, if we pre-treat these cells with inulin, notice you don't see as much invasion happening. In fact, it's like these the mortar between the cells stays intact. Similar with SC-FOS, okay? So this was quite amazing because this is now independent of the microbiome effect. This is just direct effects of the fiber. And we did see signaling effects. So fibers seem to stimulate some signaling in the gut independent of the microbes. So this kind of to reiterate that, so we found that there's a direct dampening of inflammation independent of microbes. So IL-6 will go down in response to lipopolysaccharide challenge. So endotoxin challenge, you get less IL-6 in response to that when you have inulin or FOS. Now, I mentioned that the animal-based diets, so the ones that are high in meats, these ones could produce some toxic bacterial metabolites. So back in 2014, when David et al. Had their most interesting Nature paper looking at this plant-based versus animal diet, going back and forth, they showed that deoxycholic acid will increase on this animal-based diet, and it is a known secondary bile salt known to damage DNA. So there could be conversion of this cholesterol-derived cholic acid, deoxycholic acid, by 7-alpha dehydroxylase. This is an enzyme in these bacteria that will then make this toxic metabolite not so good. So to finally leave you with last little story, which is, I think, very interesting, because I mentioned how at the beginning of this talk how the microbiome is something where is very difficult to change everybody's microbiome to the same, because we're all different, right? We're all very different. Well, somebody went about trying to see if they could make a change. So this was recently published in Cell, where Jens Walter and his team at the University of Alberta, he's now in Ireland, they tried to restore a specific microbe that they thought would be healthy that's found in Papua New Guinea. So in Papua New Guinea, there's a certain type of, it's Limosilactobacillus rhodori, this particular bacterium. It's found only, I don't want to say only there, but it's found there and not in Canadian people, right? They thought, well, if we give Canadians a Papua New Guinea-like diet, maybe we can reintroduce this microbe that, is found only there, and it would survive in a Canadian person. Does anybody want to make a guess? I'm going to ask you one question here. Do you think this could work? It's a good theory, right?
It seems plausible, but I'm going to guess it didn't work.
You're going to guess not, eh? Good guess. So yeah, it definitely seems plausible, and they definitely thought it would work. And you're right, it didn't actually. So what they did was they did on the flip side, and this is why I think this is so fascinating, is when they changed, so they did, this was like I wish I had the money to do these kinds of studies. They did a controlled feeding trial. So basically all the meals were provided for these people, like every single one of them. They had a metabolic kitchen and they did all the, meals of this Papua New Guinea style diet. And what they saw was this massive decrease in body weight, fasting glucose, fasting cholesterol, inflammatory markers, everything improved. So here's kind of a look at the food in this, it looks pretty nice, right? I mean, lots of whole foods and just, fiber rich foods, essentially. Now, they found that the bowel movements on this restore diet, they call it, improved or became more frequent. Stool consistency, got a little bit looser in the way, not so constipating, if you will. And then they also found, unfortunately, on the people with the fiber diet, they had some abdominal tension and flatulence more, not surprisingly, right? Some stomach aches, probably more so. But they did see increases in short chain fatty acids, as we would expect. And some changes in body weight, BMI, as I mentioned, cholesterol, glucose, CRP, calprotectin, and so forth. So the point is, you didn't have to get that great new bacterium that was from, Papua New Guinea necessarily to get the health benefits. The diet itself, that high fiber diet, gave all these benefits, right? These many benefits. So I think there's the malleability within each one of us. And I would say, certainly if you've, and many of us have been on, many courses of antibiotics, that's a big challenge because our microbiome has been battered pretty badly, right? But if we can gradually feed it the right things, I believe there's a certain level of resilience there that we can build back, maybe with the use of some probiotics that can come in there. But I would say the argument here is that much more for the prebiotic than the probiotic. So just to recap this, basically, putting it simply, fiber feeds these good microbes, keeps them happy. They're not going to eat your mucus layers as a result because they've got food. You've been feeding them. Fiber will feed these good microbes. They in turn feed your colon with these short chain fatty acids like butyrate, preventing colon cancer, dampening inflammation, and a variety of other good things. And then fiber also directly signals your intestinal epithelium to protect itself and further dampen inflammation. Now, I know I want to leave a little time for questions and then maybe I could do that right away. And then if we have time, I could go on because I have a little bit about like mental health stuff for microbes. If there's not too many questions. How does that sound?
Sounds good. I don't see anything in the chat yet, but somebody may have a question.
Well, maybe I could do this where should I go on a little bit into the mental health stuff and then if people can put their questions in the chat and then we'll, how does that sound?
I actually have a question, Pekka, if you don't mind.
Sure.
Yeah. I just kind of related to what you were just saying about resilience in the gut. So I'm thinking of our participants with CLIP. We're teaching a lot about plant-based diets, and people are transitioning to more plants in their diet and that type of thing. When you talked about the animal diet and how there's production of toxic metabolites and that type of thing, can you speak a little bit to say an example of where someone is predominantly eating plants and then, Christmas is coming and they eat turkey at their turkey dinner? Like, is there a resilience pattern there where the gut is, in a stronger state so it can tolerate those types of things versus if you're not feeding it that way all the time? Like, that kind of all or nothing, right? Our participants have a hard time shifting, obviously, everybody does, all the way one way. So just speaking to that a little bit.
Yeah, I think that's a very valid question. I think without obviously I'm a proponent of, less of anything bad is better, right? But that said, there's this concept called permissive flexibility, right? You maybe heard of this, where in lifestyle medicine even as well, where you say, okay, you know what, let's not be so hard on ourselves that our schedule becomes its own problem, right? Or our regimen is so hard that we become stressed as a result of that. So I think I do believe that there is some resiliency of our gut microbiota to infrequent lapses, let's put it that way, in the way that we administer our food. Now, I would say if you go back to that David et al. Paper, I would recommend looking at it because they actually, if I'm recalling correctly, and I'll have to go back and check it myself again, but I think they showed that if you switch from plant to animal diet, and like, so they were, some people who have very strict diet, like regimen wouldn't be up for this kind of study because they did it in the same people, right? Yeah, this person eating, plant-based diet, and then they just changed them completely to like, primarily animal-based. The animal-based diet seemed to kind of overpower things, maybe because it just completely was deficient in the fiber. So you just really starve those ones away. So I would argue that, if you look at something like a Mediterranean diet that still contains quite a bit of fiber, if you're not like really depleting, the fiber away for an extended period of time. And when I say extended, in a matter of days, it doesn't take long, like 3 to 5 days. What they showed was 6 days in their study. So you're probably going to be okay. Let's not beat up on ourselves about it. Let's be permissively flexible, if you will.
Yeah. Yeah, that's great. I think that helps to give, just a sense of sense of calm and peace around that, because people do struggle with that sort of black and white thinking around it, right? That it's difficult to do completely. So it's worthwhile telling them the benefits of the fiber are still there, and it's not like you're saying you're going completely animal. Still having a majority of your life filled with plants is still going to give you some protection.
Yeah, and I think, and this is where I think Mylène would touch in too, is that, calorie density, right, of plants is just so much lower. And so, you can eat, and you can really eat yourself full to be satisfied with so much plant material that, and it'll be very much less calories than if you eat, a smaller piece of meat or or oils and other processed fats. You're not gonna, I would say you can have more joy in eating if you can build that habit. That's the hard part, right? Is developing the habits of healthy eating. And yeah, whole other talk, but maybe that ties into the mental part. Does anybody else have
Pekka, I
questions?
saw Peter had a question in the chat that I also had actually. If you do a prep for a colonoscopy and they really empty you out and you're not eating for at least 24 hours and you just and also I guess the chemicals that they have you taking for that, not just the fact that you're emptied and not eating, but also whatever you're drinking, whether it's like the Peglite or Picosalax or whatever. Does that have a similar effect as taking an antibiotic, or like what kind of effect does it have on your gut microbiome?
Yeah, that's a good question. So I haven't done those studies myself. I think I've heard some. I don't want to misquote. I think for the most part, and I could be wrong on this, so don't. But I'm pretty sure that antibiotics are going to be worse when it comes, depending on the type and whether they're bactericidal or static. Because so you're going to have, even if you clean your colon well without antibiotics, you're still going to have some adhesion. Now, this is the thing, when you think about that mucus layer, if you have a healthy mucus layer before you're taking your Peglite and whatever and all the stuff that you got cleared out with, you're going to still have some adherent cells to that upper mucus layer. They're not going to be gone entirely. You're going to get most of other things out, but they're not going to be gone entirely. So that they can grow back. Like, you remember, bacterial generation times are fast. They don't take long to grow back. And so I would say that the clean out, if you started with and maybe this is a recommendation, is that pre-colonoscopy for patients to build themselves into a healthy fiber content in their diet and be in that healthy space, and then do your clean out, do colonoscopy, you probably recover better. It would be my thought.
The follow-up question was, how quickly would you recover from that?
How quickly? I think probably days. Like, I don't know.
Again, I'm assuming it would depend on what you're eating after as well.
Yeah, I think you've got to be careful there as well, right? Probably you know better as clinicians than I do, because I'm just the guy in the lab, right? So what do I know? They're basically, if you give a person heavy fiber immediately after their colonoscopy, good luck, right? It's going to be a major problem, as we saw with those individuals who are on the, on that restore diet. When you give them even lots of fiber there, and they were trying to somewhat titrate it, their flatulence and their stomach issues were pretty, painful for some, right? So
this is, however, a population that's not used to that. So to your point, like, if you've built yourself before, then it's probably very different than if you're starting from scratch.
I totally agree. Yeah, I think that's the challenge. But it might be a place, this is just a thought, because this is just a real thing you're experiencing, I'm sure, as clinicians, is maybe if you advise the patient that, okay, now that you've had your clean out, your colonoscopy and your clean out, like, maybe let's try and repopulate with good
We're
guys.
starting with a cleaner slate.
Let's start, let's build in the good ones as best as possible. Yeah.
Definitely.
So
my other question, if you still have a couple of minutes,
sure.
I didn't see if the what about fasting?
Yes. Oh man.
Sorry, did I open that?
No. Fasting is the reason I say that. So there's I think there's going to be some cool, if you look, just keep your eyes out for Bruce Valence from UBC. He's going to have some publications pretty soon. I don't know if they haven't even already come out, primarily looking in mice, but I talked to one of his postdocs, I think, or PhD students at a conference, and they were looking at just a 48-hour fast in mice, which is actually pretty strong for a mouse. But they see a dramatic shift in the microbiome and also in the mucus layer. And it's not negative. Let me just put it that way without giving it all away. So fasting definitely has benefits. And however, you don't want to starve your microbial self, as I've already said. So I think that, an intermittent fast periodically isn't going to do that. It's kind of maybe goes back to the initial question. Okay, you're going to lose some of those microbes, but maybe you're going to reset to some that are, are more dependent on the fibers if you come back with fiber after. So yeah, I think it can be a good reset mechanism for sure.
Is there if you do like 12 to 16 hours every day is better than, I know that some people do like 24-hour once a week, 48 hours. Some people go on fasting for like 4, 5, 6 days, 10 days.
Yeah. Yeah. So that, I think that, some will argue, that time restricted feeding. In fact, I'll go forward to this slide because I was going to show you HASCAP pictures. Those are HASCAPs. Just so you get to see them. They're kind of like long blueberries, and they have a lot of anthocyanin. So, 3 to 4 times the amount. But just to quickly here, I want to get to this slide. So notice, leave at least 12 hours between last meal and first meal. So this is from the I forget, Zoe Science and Nutrition. That group that looks at
Dr. Will,
it's pretty, Dr. Will, yes. So basically, they have shown through a variety of large-scale studies that for benefits of your microbiota, having that 12 hours, especially at night, at least. Notice you could be 18 hours. I'm not saying I practice this, but I wish I did. If you eat your last meal at 3 or 4, and then you eat again at, first thing in the morning kind of thing, you're going to be better off in terms of allowing. Part of the reason for that, I think, might be also just allowing the microbes to, maybe some of the ones found higher up in your intestines, to grow a little bit and maybe produce some vitamins that we might like to have. I'm thinking B12, and I know that might be controversial, but it may be that giving them enough time to just sort of be there and produce some things could be actually helpful. So yeah.
That's super interesting. Yeah, because I mean, our bacteria producing some of our vitamins is also something that's cool.
Well, they do. Yeah.
I just wanted to have 4 plants on my
Peter, you've got some
property.
haskaps? Awesome. Are they
Nice.
growing? Have you got any berries yet? Maybe a few.
There's a lot in Quebec. There's like
Yeah.
fields of them. They're called
Yeah.
camaries.
Camaries. Yeah, that's right.
Yeah.
Camaries. Yeah.
Yeah. So
Yeah.
it's
Yeah.
which
Yeah,
is
it's a big thing.
interesting that I never knew about them until I went to your conference, but I guess where I grew up there's lots.
Yeah.
I think Kathy is messaging me that she somehow got kicked out of her own
Oh
meeting.
no. Oh no.
But we'll keep an eye if there's any other question, if people want to unmute themselves or pop them in the chat.
Yes, by all means. Yeah, and I can go back and show you the picture of the haskap berries too. So we did do a food-based clinical trial with these. And yeah, if any questions come up, please don't hesitate to jump in.
And they're delicious.
Yeah, they taste really
They
nice.
stain like nothing else though.
Yeah.
Well, if nobody else has, I would love to hear what you have to say about the connection between microbiome and mental health.
Okay, well, let me, I know our time is, I don't know if I have to cut it off right at 11. I'm happy to talk for a second, but if somebody wants to stay,
I mean, if people have to go, there will
they
be
can
a
go. Yeah, I won't be
replay that's
offended.
available, and
Yeah.
and I don't know if you want to maybe pop your email in the chat if some people want to
Oh,
reach
yeah.
out.
Yeah, I put my email in there, so if anybody has a question, they don't hesitate to send me a note.
Amazing.
So yeah, I'll go back to talking about mental health here just quickly. So we know neuroinflammation is linked to a variety of different psychiatric disorders, appears to also be influenced by gut inflammation, not surprisingly. And so when we think of our brain immune mediating cells, if you will, our microglia, they seem to be the ones that are sensitive to this gut inflammation. And if we administer short chain fatty acids, or which could be coming through circulation, they can help to dampen this issue of microglial dysfunction. So there's been some studies showing that. And this idea of dysbiosis, and again, I think the definition is one, you need to be, dysbiosis, okay, there's an imbalance, there's some microbial community that's not healthy, has been linked to anxiety, depression, neurodevelopmental disorders, so autism as well, neurodegenerative disease. Now, these are links, these are correlative, but certainly there's quite a bit of evidence suggesting that there are some links there. So we don't have causality yet, but just try to hide that for a second. But the studies in animals are pretty compelling. So when we think about the gut microbiome, brain axis kind of thing, there's a lot of possible interventions here. Now, when you think about diet, serotonin, which of course is an important brain molecule, very critical, right? Dietary tryptophan is really the precursor for that and raw material for that. But it can also be metabolized into chynurenic acid or indoles. So how this happens, so if we look at serotonin production, so there's this modulation of this process through gut microbes. Production of indoles is dependent on gut microbes. Serotonin and kynurenine, they're not necessarily dependent, although they're modulated by gut microbes. So how does this play out? Well, if you have lactobacilli, which are some of your healthy ones, they can actually inhibit tryptophan metabolism to kynurenine. So why is that good? Well, then you have a buildup of more tryptophan. So, and that can help maintain levels of tryptophan so that you can convert it into serotonin. So it's kind of an artificial, I don't want to say just a serotonin boost, if you will, to have this lactobacilli inhibiting the metabolism to kynurenine. There are, so indoxyl sulfates, these are specific indole metabolites made from indoles, produced by certain bacteria that have been linked to some kidney neurotoxicity and some other neurological conditions like ASD and so forth. And whoops, I went too fast here. If you have a high animal protein diet, you get more of this indoxyl sulfate, paracristol sulfate, and these ones they've been shown in some studies to have effects on those conditions. What about some of the yeasts? So if we look at Saccharomyces cerevisiae, high levels of some of these fungi, which respond really well to sugars. So that's been making the social media rounds, this like, Alzheimer's is just type 3 diabetes or something, right? Well, high sugar content in our diet can definitely have an effect on our, as of course, from Candida and so forth, it can influence the growth of these fungi. Well, can that be having influences on our brain? Well, there seems to be at least some correlations between levels of antibodies against Saccharomyces cerevisiae in Parkinson's, schizophrenia, and then also in bipolar disorder. Candida also seems to be high as well, the antibodies against those. So there seems to be some effects of some of these things. Now, high quantities of dietary simple sugars can lead, as I mentioned, to increases in Candida and lower short chain fatty acid amounts. So this is where I try to dive into the colorful fruit phytochemicals. And I know our time is short, but just because we talked about HASCAPs, I'll mention it to you anyway. So these phytonutrients are, I would say, really need to be considered nutrients. And I think newer nutrition textbooks are recognizing this. They have so many bioactive components to them. If they're not absorbed by the colon, they can actually be metabolized by bacteria into helpful things. And that's where the microbiome gets involved. So here's just a few examples of them. Urolithin, so where do you find them? Berries, walnuts, pecans. So this can help prevent neurological disorders, apparently even promoting cell death and leukemia in some cell studies. Sulforaphane, you may have heard of this in broccoli or broccoli sprouts. So this one has abundance of anti-cancer activities, also its metabolites as well. And then turmeric, you probably have heard of, pretty potent anti-inflammatory, has a lot of evidence of its effects on protecting microglia as well. And then we have onions, grapes, berries, cherries, broccoli, citrus, high in this 3-HPPA, which is a quercetin metabolite. And this is having a variety of, for example, inhibition of alpha-synuclein being the one involved in Parkinson's. So not necessarily clear causal connections, but the one I'll focus on here quickly because, of course, HASCAPs are my interest. So anthocyanins are super high in HASCAPs, about 3 to 4 times the levels of antioxidants overall in HASCAPs compared to blueberries. Here's a look at the berry again, picture of it. They really taste quite good too. I think anybody who's had one, they're definitely not as sweet as a blueberry, but definitely as juicy or more and potently staining type juice. And so what we did with those berries, just because I'll just mention this quickly, is we did do a food-based clinical trial with smoothies, 140 grams of berries every morning. That hasn't yet been published, but we're pushing that out pretty soon here. And we did see some benefits in combination with other whole foods. So whole ground flaxseed and actually oat milk. So it wasn't on its own like this amazing, wonder bullet. And I would think that that's the case for most foods. We're not going to change somebody's life and lifestyle and health just by giving them one food. Typically, that's a holistic thing. So just as my final conclusion here, I like the Zoe Science and Nutrition improvement of microbiota, sort of big principles, if you will. Don't count your calories, right? Enjoy your food, eat lots of different varieties, consume fermented foods, eat different colors of foods I just mentioned. Leave at least 12 hours between your last meal, as we talked about. Avoid these blood sugar spikes. So not too much processed sugars, 30 or more different whole plant foods per week. So they came up with this idea based on a lot of their microbiome research, and then avoiding ultra processed foods. I think these are all really great principles. I would say I'm not sure I buy into the idea of like getting your microbiome tested based on what I've said before, that it's going to tell you like this is you've got a good one or a bad one. But if you follow these principles, I think your microbiome will be healthier. So any more questions? I know we've gone over time, but I think we'll have to leave it there.
Okay, I'm not seeing anyone popping up. So thank you so much, Pekka, for coming and sharing your knowledge with our team. And I know there was some good conversation. I missed a bit of it, but thank you so much for coming. And yeah, I look forward to hopefully meeting you one day if you ever come back to this area of the country.
I will definitely come back. It's my favorite place to haunt is Georgian Bay and Franklin Island. So love it out there.
Thank you so much, Pekka.
And I'm sure you can get people to do an evaluation post afterwards. Stuff. So
yeah, I send out a survey, so everyone knows to look for my survey.
Appreciate it. Thank you so much. I'll look forward to the feedback, and thank you all for listening.
Thank you so much. That was so great. Thank you.
Awesome. Take care.
Take care. Bye-bye.
Just
yes.
All right.